And it's time. And it's time. Okay. Okay. Good. Well, we're getting back to mass spectrometry at this point. We talked a little bit about different sources of ionization, how we blow a molecule apart, where we get the energy from that. A little bit about how we separate also then these fragments, these bits and pieces that the molecule's been blown into, and how we measure them. So we kind of got the parts of a mass spectrometer. But we do like to interface these with tools that do separations for us. When we start putting mixed compounds into a mass spectrometer, our data are a bunch of mixed fragments, and it's hard to say where they come from, and there are multiple molecules. So we really like to tie a mass spectrometer into some means of separation. So let's separate a mixture into components and then get our mass spectra so we get each component. Much better success in terms of identification. And so we were looking at a gas chromatograph. Gas chromatograph, remember, it's has a mobile phase. It is a gas, and that gas is fairly low in volumes. And so we think, what's if I had the number down there? Maybe I don't have a number. But anyway, we can, because it's such a small column and such low flow rates in that column, the mobile phase, we can actually just do a direct interface. In days gone by, or maybe I should say early days, we had much bigger columns. Much bigger columns meant much faster gas flow, and the mass spectrometer could never pump that gas out. It just couldn't deal with it. Now, like I say, with very tiny columns, small columns, we can just directly interface it. Take the effluent out of a gas chromatograph, and hopefully one compound by compound comes into the mass spectrometer for us. We still have to have darn good pumps. And so we have what we call a turbo pump, a turbo molecular pump. And that can get us down into the region we like. We like to see about 10 to the 4th torr inside of that mass spectrometer so we can get good data. If we don't have good vacuum in the MS system, basically, all the ions we just formed, the bits and pieces, will react with air and they'll be neutralized and gone. So we absolutely have to have a good vacuum, and that is accomplished, like I say, in this manner. So like I say, turbo pumps is what they're called. And the idea of what kind of volumes are we pumping out with these pumps? Well, 1 milliliter per minute of gas coming out of our gas chromatograph, so out of the column, turns into a volume of 10 to the 6th mils per minute when we get it under that high vacuum. Obviously, the vacuum air expands, volume expands. So, like I say, we really depend upon good vacuum pumps. It's fascinating to me and maybe somewhat irrelevant, but we have these turbo pumps, and we've got machines that turn about 55,000 RPM. That rotor inside is rotating 55,000 RPM, and they've run over 30 years. So, you know, these pumps are really built well. They're very robust, and they have really good lifetimes. But good vacuum pumps are the heart of the system. If you don't have good vacuum, you don't have good data. And so that is the gas chromatographic system. We've got a tiny column here, not a bigger one as it shows here. Interfaces directly with the mass spectrometer, the quadrupole mass spectrometers you'll see there. And that gives us our system. Now, I didn't go over this figure? Yeah, that's what I thought. Okay, so let's, how about this one? I think we started a little bit earlier maybe than we needed to. So anyway, you got a double dose perhaps of gas chromatography mass spectrometry. So here's when we start interfacing with HPLC, high performance liquid chromatography. Typically we call it LC these days. And in this case, that mobile phase, that column is much bigger, and the mobile phase is a liquid. And so that liquid is going to turn into a gas in extremely large volumes of gases. So it is really a problem. We can't just directly put an LC column effluent into the mass spectrometer. We have to have a system where we get rid of that solvent. And that's what we showed a little bit earlier, the electrospray ionization. In that particular system, this is another diagram of it, you've got our LC column coming in on your left to the ESI nozzle, electrospray ionization nozzle. The center of that nozzle has about 4,000 volts of current at it. And that's so much current that it tends to ionize the molecules of the liquid and of your sample. And so it gets a spray droplet. In that spray droplet, you have ionized compounds that you're interested in identifying. It's run under a situation where that solvent evaporates. So as it comes out of the nozzle, the liquid solvent is volatile. It will evaporate. And it will leave us then just with our ions. And the inlet, where it says heated capillary up there, that has a negative charge on it. So they tend to draw those electrical positive ions into your mass spectrometer. And they go through the separation and measurement. So it's a really robust system and a highly successful, highly used system. I do something a little bit different. I'm interested in what's in the gas phase. So this type of source here is called an atmospheric pressure chemical ionization inlet source. It's somewhat similar to what you just saw for the ESI, but a little bit different. In that case, we don't have a liquid. This type of mass spectrometer would be up in a balloon, for example, in the atmosphere, monitoring pollution or following buses through a metropolitan area. So it would be looking at air pollution as one example. I look at it for flavor compounds that are in the air that we smell. But in this particular system, we've got a capillary inlet that's forcing some gas or sampling some gas in it. Right at the end of that, where it says corona needle, that's about 7,000 volts. So right here, we got 7,000 volts. So whatever comes in through this capillary in our gas streams being drawn in, 7,000 volts, it basically ionizes the molecules that we want to measure. It's drawn into the rest of the mass spectrometer, so it gets drawn in here, into the mass analyzer. They get separated here into fragments, whatever fragments there are. There aren't many fragments, it's pretty much the parent ions. And it gets measured by the mass spectrometer. I'll show you an example later on how we use this particular system. When you get a mass spectrometer or a mass spectrum profile out of a LC or a GC, you always see, of course, on the top, you've got just basically the total current, the number of fragments, number of charges that are coming out at any given time. It draws a line on a piece of paper or draws, a line on your monitor. That doesn't tell you much, but the whole time that basically you're sampling the ions in there, and we draw this curve. Here's compound, here's another one, so on. The machine is doing its job. It's taking samples, blowing them in bits, measuring, blowing them in bits, measuring. They can do that many times a second, hundreds, maybe 1,000 times per second. And so the whole time that it's drawing this line, it's getting separate mass spectrums over each of those peaks. And so that's kind of the image that's shown here. So we've got basically different spectra coming across as a function of time of your LC or your GC. And so in the end, you'll take these individual spectra, the profiles, and do the computer searching on those. And so the computer will try and match fragments to some known compounds and tell you what that compound is. So, and that's one technique, is looking for identification. That's one of the major jobs. But it's also really good for quantitative analysis, how much of something happens to be here. In some cases, we want to, let's say, get an accurate measurement of a compound. Again, I might be looking for some flavor compound or some off flavor compound. And so what I want to do, if I want a really good quantification, I will spike my sample with an internal standard. Now that's where the fun begins, because how do you choose an internal standard for your analysis? If we had a good live audience and a lot of discussion, we could go through all kinds of thoughts about how you choose an internal standard. Have you spoken about that in earlier? Sure. And one of the basic principles is you want that compound to be like the compounds you're going to measure, so they behave the same way. If there's any losses, okay, that would be reflected in the internal standard, any poor extraction, whatever happened to be. So you really like to have an internal standard that's as close as possible to the compounds you want to measure. And there's really nothing better than using an isotope of the compound you want to measure. And I'll show you an example of that coming up, of how we can do this. So I'm not using some related compound. I'm interested in cherry flavor, for example. That's benzaldehyde. Okay, so I'm interested in measuring cherry benzaldehyde. So I'll take benzaldehyde, I'll put a carbon one bigger, C13 into it, and then now my new benzaldehyde, my standard benzaldehyde, will have one more mass than the benzaldehyde in my sample. And I'm going to then look on the next plots and see how I can measure the heavier mass. So we're using, like I say, the same compound, just made it one mass units larger, and then we can go ahead and quantify. And this is basically, in this case, they haven't added a carbon 13, they've added a nitrogen 15. So their standard is one atom bigger. And what is this? This is dimethyl derivative of 5-fluorouracil. So, okay. So you can then have your sample. You've added your isotope to it, your sample. You run the sample and you tell the mass spectrometer, measure only the mass that's going to come from this compound. Okay, that's our compound of interest. We're going to measure only that peak. And then right underneath it, we're going to tell, okay, and look for the one mass unit bigger. And that's what we have here. Here's your N15 standard, and your one right above is your nitrogen 14 sample. So it's a marvelous internal standard. There's just no better way to do this than this type of analysis. So spiking with labeled isotopes. Purity of a GC peak. You know, whenever I read a publication, somebody shows me a chromatogram from an LC or GC, I always say, okay, what is that? Oh, that's this compound. How do you know it's that compound? How do you know it's only that compound? I think I mentioned that flavors, aromas, we have over 11,000 aroma compounds. I'm not going to separate 11,000 compounds in my GC, no matter how good it is. There's a good chance that some things are not being separated. And if I report them incorrectly, I'll say, oh, it's this, and there's this much of it. I'm wrong. That could be devastating. You might be doing detection or limits of some carcinogen in food. And if you don't know what you're measuring and you're measuring only that, you could be in big trouble. So you really like to use mass spectrometry to say, this is it, I measured it, and that's all I measured. So in that case, what you do is you run your mass spectrometer across each peak and see that it's the spectrum, the pattern only for that compound and only that compound. We have the ability to do something that's pretty neat also. And I'm not stuttering, it's not MSMS. It is MSMS, I'm sorry. It's not a mistake. what we do is we do mass spectrometry. We blow this molecule into pieces, and we get all those pieces. Oh, that's nice. But then we blow every one of those pieces into more pieces. Because if we have a mass someplace in there, a fragment at, let's say, 128 mass, gosh, that could be put together of different things. How do we know what makes that 128? So let's take that fragment, blow it to bits. Oh, okay, that's what it is. So we know that the fragments are from a specific molecule. It's really ensuring, again, that you're measuring what you think you're measuring, and absolutely. So we actually put two mass spectrometers together. We first blow the molecule to bits, and then we take each of those fragments and blow them apart again. So we can look at the fragments of the molecule and the fragments of the fragments. So really a good way of getting absolute quantification. And this is what we call the MSMS. It's basically, there's two quadrupoles in this situation, two sources, two quadrupoles. So you just couple them together in a system like this. Cost, they're expensive, $250,000, $300,000 easily for this type of instrument. You can go into much more money than that, but they are a triple quad. They're really a useful addition to us today. These are some applications. I gave you horse or cobra venom for horses or whatever as on first day about applications of mass spectrometry. Well, okay, here's something a little bit closer to home. And I'm going to give you an example of flavors reacting with proteins. I'm going to talk a little bit aroma release in the mouth. You know, when you put something in your mouth and you taste it and you smell it, how do you know what really came out of that food and was there for you to smell, went through your olfactory region? Did most of it go into your stomach? You swallowed it, put it in your mouth. How do you know what really came out of that food? And so this is where we get into aroma release in the mouth and mass spectrometry. I'll give you an example. Identification of unknowns, assurity of purity, quantification, adulteration. Something's there that shouldn't be there. Somebody swapped out some ingredient. And so again, mass spectrometry, a way to look for adulteration in foods. And this is one that I like, obviously, that comes out of our lab, basically. And your advisor here gets credit. And so this is looking at beta-lactoglobulin. That's the major whey protein in dairy products. So beta-lactoglobulin. If you take a look at this, this is one spectrum. If we would go over to the first peak here, that is a mass of 18,276. That's beta-lactoglobulin intact. It hasn't been blown apart. That is the molecule just given a charge. If we take a look then the other peaks that happen to show up along this line, we've got a few oxidation products, but interestingly, we've got beta-lactoglobulin that has a covalently bonded lactose. Gosh, a reducing sugar reacts with a protein. What does that sound like? I hope it sounds like the Maillard reaction to you. Yep, a normal reaction in foods. Obviously, milk contains lactose. There was an opportunity for this reaction to occur. And then we've got one more molecule down here added on. So this is two molecules of lactose added on to that protein. So that's just our protein that happens to have reacted some with sucrose, with lactose. Now this is where the grad student who did this put a little bit of cherry flavor in there. Okay, now we got beta-lactoglobulin that has cherry flavor added to it. And that is, like I say, benzaldehyde. The figure on your left over here, that's pure beta-lactoglobulin. This is, whoops, we went the wrong direction. And this is a new peak. It's 18,276 plus the molecular weight of benzaldehyde. Here's our beta-lactoglobulin with one sugar added to it. Okay, and here, right next to it, is another red arrow of another benzaldehyde. We can actually show that we're adding flavor onto that protein. that's really relevant to us in the sense that proteins are hard to flavor. One of the issues certainly the industry is finding is that when we add more protein to foods, different types of proteins, the flavor doesn't perform well. It's pretty good on day one, but gosh, 2 weeks into shelf life or a month into shelf life, that flavor's changing. It's going away. Where is it going? It's covalently bonding onto the protein. It's a shelf life problem. It's also a storage problem because if we start, I'm sorry, not storage, it's a processing problem. We start thermally processing this. Again, very often some company will call and say, gosh, you know, I just put this through a high temperature short time, or I just sterilized it for an aseptic package. My flavor disappeared. Where did it go? It added on to the protein. So it's interesting. What can we study? How can we learn from it? How can we make this better? This is after the proteins had 6 hours to react. And we see again the red arrows up there, where that peak is now not small. That peak of adding the flavor onto the protein is almost as big as the protein was. So fascinating area. We couldn't do this without mass spectrometry. We would never have a way to measure this without mass spectrometry. It's a way to solve a problem in the industry. This is another one is an example, just illustrating the variety that comes out of this. And I don't know, if you know, we could just have a show of hands. How many of you love diet beverages? How many love sugar sweetened? I am personally a diet beverage. I like it. That's preferred. I drink it for that reason. I don't give a rip about calories. And there's a lot of question about that these days. But I just like the flavor. Why are they different? The company originally starts off putting exactly the same flavor into water that has sugar, and they put it into water that has aspartame, for example. Why are they different? Why should they be different? And so, again, mass spectrometry is a tool that's been used to look at this type of issue. In this particular experiment, added flavor to a non-sweetened beverage base, aspartame sweetened beverage base, and a sucrose. So we got 3 different samples. And so we were interested in what's given up during the drinking process to go back to your olfactory region and say a flavor sensation or get a flavor sensation from it. And so what do we do here? This is a fellow, it's one of the researchers. If you could better see that perhaps, you see there's a little glass tube going up his nose or his nostril, and the mass spectrometer is drawing in his breath. It's continuously monitoring, continuously taking spectra every time he exhales. He puts the product in his mouth, he swallows it, he exhales when he swallows, and he goes on breathing and exhaling, inhale, exhale, and the mass spectrometer measures what was given up from that food for sensing. And is there a difference then between aspartame, water, and sucrose? Can we start trying to understand why there are these differences? Now this one I hope you don't get lost with, but so I'll try to, let's say maybe a little bit easier, a little bit briefer. So this is the spectra of his breathing. All living bodies give up acetone. It's part of our body's metabolism. So every time we exhale some acetone. It's part of our metabolism, okay. And so these bumps up here, the red ones up here, they basically are measuring his breathing. That line goes up when he exhales, it goes down when he inhales, and so on. And then all the rest of these other lines that come across, so your second, oops. Each one of these are measuring different flavor compounds. So the green one, the purple one, and so on, and each of those are looking at other compounds, flavor compounds. When he swallows, there's a big bump. It's called the swallow breath. Lots of aromas given to the olfactory region. Then by the second breath, third breath, it's basically gone or pretty much gone. However, some compounds in there, they continue to come along. So some of these middle ones in here, very volatile, they go away. The bottom ones, they tend to persist. So it starts telling us something about how we perceive things, what influences how we perceive flavors. So it's, again, like I say, just a fantastic tool for those kinds of purposes. So those are two things that we use the mass spectrometer for in the flavor group. But again, the idea of purity, if we were doing any kind of toxicity work, if we were doing any work we really want to measure something accurately, know what it is, this is, as I said, we look at each peak, we measure those peaks, and we determine they are pure peaks. Adulteration, ID adulterated, pesticides, contaminants, off odorants, and so on. So the identification of these types of things, again, mass spectrometry does wonders. This is only, there's a couple more examples only, but again, I'm so impressed with creativity of people. And so what is done here is that actually it's an animal brain, not a human brain. Don't remember what it's, which one, but the brain has been, obviously the animal died. You cut the brain, what's that? Mouse brain, you're right, okay. I see it is kind of a big brain, but yes, okay. Anyway, so you can then do mass spectrometry of cross sections of the brain and look and see how the brain structure has changed, how the chemistry of the brain structure has perhaps changed with disease, changed with diet, changed with age. So we're actually going to do pictures. We're going to get pictures of chemicals reacting in the brain. For my part, I'm a little much more mundane than that. I'm interested in encapsulating flavoring materials. Flavorings are volatile. They're going to evaporate. I want to protect a flavor, so put it in a dry powder so I can put it in a mix someplace, and then the consumer can add water to it and get the flavor back, right? Think of all the dry mixes on the market. Well, the only way you can preserve a flavor in a dry mix is to put it inside of a capsule, inside of a particle. And so basically, that's what we do out in a pilot plant. We spray dry flavors and we protect them. But it's interesting, what is the structure of that particle? And so this is a technique where I can look at that particle and see how it changes as water evaporates, as flavor moves out of it. I can do mass spectra and get an idea of structure of that particle. I can better understand how to make a better structure, a less permeable structure. I want to trap it there, tell it dissolve it, and have it released. And so we have scanning mass spectrometry that has a limit of some, I think it's like 2 angstroms. It can look virtually at molecules across a powder. Oh, question, where's the sodium? Okay, that's a kind of a different question. But it is an amazing technique. It absolutely fascinates me all of the ways this is used today and how it's evolved throughout time. It touches us in so many ways. At one time, this was only for the elite laboratories, experimental laboratories. Cost has come down a great deal. So instead of maybe spending, $400,000, $500,000, I might be spending $100,000 today. It's brought it into the hands of everybody. That's a very reasonable price for what you get. They are easy to operate. They are basically push-button systems. You put your sample in, you get an answer. This is the right answer. That's up to you to make sure. But you will get answers. So it doesn't take a PhD with 50 years of experience to understand it either. So prices come down. They're easy to operate. Computerized databases, so searching's done for you. And so this whole idea has really brought mass spectrometers into many food labs that they wouldn't have had before. So it's impressive where it's gone. Oh, now your advisor gets to take over. Yep. Anyway, if you ever have any questions about mass spectrometry or these kinds of things, what I've talked about, whether it's GC or LC or MS, I'm up in 147 in the emails. I'm always happy to spend some time with you, whether it's now or after you graduate. I'm an optimist.
Okay.
Thank you, Pam.
Thank you,
So, have a
Gary.
good day. Enjoy.
I'm gonna let them go.
You're going to let them go.
Yeah, I'm not gonna start the topic now. They will hate me.
They look disappointed, actually. I think you should.
I think they're gonna appreciate going working on the lab reports. Okay. All right, guys, I will end here today.
Okay.